All right. Now it's my pleasure to introduce Kritika. And Kritika actually started this last fall. As I mentioned, Kritika started a fall a year ago and did a project, and that's going to be tomorrow. And this was a new project she was working on here. Kritika just started this last fall, and she was working with Barrett Hester, who's not here to present. She's presenting the work that they worked on during the fall. And she's continuing on in the labs, continuing to get some more research going where she's interested in. But she's presenting this preliminary work from the fall on methylene blue.
So good afternoon, everyone. As Dr. Briegman mentioned, I am a freshman undergraduate, and I had been shadowing this research project for the last semester, and I'll be presenting on the utilization of Xenopus embryos for the determination of the teratogenic potential of the methylene blue. I know it sounds a lot, but trust me, we'll go through it. So, being in the lab, the mostly I learned was to just learn the basics of setting up a research experiment and learning how to get used to working in a lab and learning microscopic images and setting up frog embryos, collecting them, sorting them, and I just got an opportunity to learn a whole lot about the frog embryos. And as a result of this, I would like to present. So this here is a time lapse of the first 48 hours of a Xenopus frog egg. And as you can see that the cells are continuously dividing through mitosis, and which leads to the further development of the entire frog embryo. And as a result, we have a frog embryo. And it was done through a microscope camera built into it, and it just took a series of pictures and we just put it into a time lapse according to the frame. So now, moving on to our experiment. So the main objective of this experiment was to test the potential toxicity of methylene blue on these embryos. So for this, we decided to perform the experiment in PTAX. So what is PTAX? PTAX is the 96-hour test performed on the early stage embryos of South African clawed frogs, also known as the Xenopus frogs. So in this experiment, in PTAX, what we mainly do is we determine the potential of a certain chemical or substance in causing malformation, mortality, and growth inhibition in these early stages of the embryos. So the main reason behind using these specific frog embryos is because these embryos develop outside of the body, so it makes it easier to manipulate them with chemicals and proteins and to witness their effect of these chemicals directly. PTAX has also been approved by the Environmental Protection Agency as an assay for the identification of any other human intoxicants. Now, moving to methylene blue. So why did we decide to test on methylene blue, and what is methylene blue exactly? So methylene blue is a dark green crystal, or it can also be found in a crystalline powder form. And when it's mixed into alcohol or water, it gives out this very vivid dark blue color, which can be used as an indicator or a bacteriologic stain. So in recent studies and throughout the history, methylene blue has been considered a safe drug, and it has been used to treat early stages of malaria throughout the late 19th century towards the beginning of the 20th century. And it has been used to treat methhemoglobin until to this day, but after a certain amount of the safe doses and the concentration, methylene blue is known to be highly toxic, carcinogenic, and non-biodegradable. So it has adverse effects on health and the environment. And studies have also found that methylene blue can cause severe nervous system damage and nitrogen and ammonia poisoning. So to figure out how severe these effects are, we set up an experiment with the Xenopus frog embryos. So what we did was we exposed the Xenopus embryos to a range of different methylene blue concentrations under laboratory conditions, and the concentration ranged from 0 to 500 milligrams per liter. And for each trial of the experiment, we set 4 controls to see whether or not these changes that we were experiencing was due to methylene blue or something else. So this is what our experimental setup looked like. So in each petri dishes, we had a different concentration of a methylene blue. And in each petri dish, we had 20 different embryos sorted into it and 8 mLs of solution. These solutions were changed every day, and the number of embryos in each petri dish were measured every day to note down the number of casualties. And we also had the controls set up with only embryos and fetal solutions. So after the 96th hour of the test, we decided to take certain analysis of different kinds of factors. So one of them was length measurement. So we decided to take pictures of the Xenopus embryos, and we used those images to find out the length measurements using a computer program known as ImageJ. So that program enabled us to measure any object that is within the picture using the scale that's also been present in the picture. So as you can see, there's a scale that we provided through which the program could use the length measurements. And it would be generated into a spreadsheet data. So now moving on to our results. So the blue line represents the mortality, and the orange line represents the malformation. As you can see, the mortality started to increase when we raised the concentration of the methylene blue, so much so that at the highest concentration, which was 500 milligrams per liter, our mortality rate was up to 100%. And same with our malformation. The malformations were seen more often when we started increasing the concentrations of methylene blue. Now this is very interesting to see because it further provided evidence that methylene blue has a certain growth inhibition effect, as we could see that the length of the embryos were shorter in higher concentration of methylene blue than in comparison to the controls and the lower concentrations were. We also, after the experimental takedown, we also put petri dishes under the microscope, and we observed each of the embryos and saw what kind of potential abnormalities that we could notice. And these were the list of the abnormalities that we saw, and it was like a damaged tail. So usually a normal frog embryo would be straightly aligned. So if it is bent or coiled like that, that means that it has a body or a tail abnormality. And gut abnormality, it's not that clear in the picture, but usually in Xenopus frogs, the gut is tightly coiled. So if it has a loosely coiled gut, it also gives out this larger appearance, which suggests that it has an abdominal or gut abnormality. And edema. Edema are swellings in different parts of the body due to high salt concentration or just like injury in the body. These are the actual abnormalities that we were able to document in our research. So first is an underdeveloped embryo. So this embryo was highly malformed, and it could not go through a proper developmental stage. So it probably had a very severe effect of methylene blue. The second one was the most common one, and it was like a bent tail and a nodopart damage, in which a lot of embryos were noticed to have a malformed tail. And the third were blisters, where they were usually found around the region of the head. So to summarize our conclusion and finding of this research, so our LC50, which is the lethal concentration 50, in which we would assume that half of the population of these embryos would be dead. And it was found to be 256.41. And our effective concentration 50, which is the malformation, in which we would assume that the half of the maximum potential of methylene blue could be seen. And it was found to be 135.63. So in the studies, it has been figured out that if the ratio of LC50 and EC50 is more than 1.5, the chemical or any substance would have an increased teratogenic risk. And our experiments showed that it was 1.89, so we could conclude that methylene blue had certain amounts of teratogenic risk. And we did find abnormalities in other different parts of the body, like sometimes the embryos would be stunted, they would have a damaged eye, or like notice conditions as hemorrhage and other kinds of blisters. And in total, we were able to conclude that methylene blue does have a certain amount of toxic effect on the development of the frog embryos. So I would like to thank Barrett Hester, who's not here today, but he was the senior student who I worked this research with, and he performed the first 2 trials of this research. And Dr. James Rayburn for being such a great mentor and allowing us to use the lab. And obviously the Department of Biology for providing us this opportunity and such a great learning experience. So these were my references for the presentation. And if you guys have any questions or queries about my presentation today, please let me know.
Would you go back to the slide where you had the graph of growth
Yep.
and its effect? Because you were saying that the EC50 was 135, and so things really pick up after that 100. But the slide after that, you showed growth, and it started having an effect at much lower concentration. Is that right? So like the one before that compared to this one was really interesting.
The 20th one?
Yeah. You're saying even at 20, we're starting to see that growth drop off
Yeah.
hugely?
So since it's a preliminary research and we have yet to perform more trials of this experience, right now we are considering this as an anomaly, like because it does show a gradual decrease in the length, but there's a certain spike at the 20th one, and we need to do more research in this and run more trials to figure out the exact effect of the length measurements.
Oh, but even this huge drop is 5% roughly growth? Is that how much smaller it is?
Yep.
Okay. Other questions for our speakers?
Just a comment that cell growth video was very interesting.
Yes, I agree.
And I'm sorry, since you mentioned that, who put that together?
Oh, wow, you made that video?
Yes.
I figured
It
you
was
just
like
found it.
a compilation of 4,000 pictures of frog embryo in a 20-second video.
Wow, that is very nice.
Wow. That was awesome.